Hello, everyone. Thank you for joining us today. My name is Mike Mitchell. I am the Business Area Manager for Eurofins Discovery. Today, our webinar is going to be about screening approaches for reducing attrition due to drug-induced liver injury, or DILI, early in drug discovery. So our presenter today is going to be Yang Zhao. He's the Director of our in vitro toxicology services at Eurofins Discovery. Our abstract for today's presentation, we will talk about DILI, as it is the leading cause of drug failures in clinical trials and is a major reason for drug withdrawals from the marketplace. DILI is a very challenging issue within the industry, mainly because DILI is multifactorial, meaning that there are multiple components that can cause DILI in the clinic. In this webinar, we're going to talk about some of the in vitro assays that we use to tackle these challenges for assessing DILI preclinically and clinically. And we're going to discuss some of the newer DILI screening approaches that we've developed at Eurofins. And at this point, I'd like to introduce Yang Zhao and have him present. Thank you.
All right. Thank you, Mike, for the introduction. Thank you for attending my presentation. My name is Yang Zhao. I'm the Principal Scientist and Study Director for In Vitro Toxicology Studies at Eurofins Discovery. I have been managing the in vitro toxicology services at Eurofins Discovery for over 15 years. Today, I'm going to talk about the in vitro screening approaches for reducing attrition due to drug-induced liver injury, in short, DILI, early in drug discovery. And in particular, I will talk about what in vitro assays that we offer at Eurofins Discovery to de-risk your drug candidate's DILI liability. And this is the outline of my presentation today. First, I will talk about the background of DILI. Why is it important to assess DILI risk at early stage of drug discovery? What are the causes of DILI? And what are the typical strategies that pharmaceutical companies utilize to assess DILI risk using in vitro assays? At Eurofins Discovery, we have developed and validated two panels of in vitro toxicity assays to predict DILI risk. Those are DILI screening panel and the DILI magnetic panel. I will talk in details about those assays in the two panels and the utility of the panel in early drug discovery process. First, let's ask a very important question. Why is DILI assessment in early drug discovery important? As we know that drugs are the major cause of liver injury. More than 900 drugs, toxins, herbs, and dietary supplements have been reported to cause liver injury. Approximately 75% of the idiosyncratic drug reactions result in liver transplantation or death. DILI is also a major attrition for drug failure in preclinical and clinical trials, which results in tremendous loss of investment and time in drug discovery. DILI is the most common reason cited for withdrawal of an approved drug from market. An analysis done by the Organization of Drug Discovery Data in 2018 found that among 192 drugs which were approved but subsequently withdrawn from the market worldwide, hepatotoxicity is the leading reason for attrition. It is estimated that the average cost to bring a drug to market is $2.6 billion and takes over 10 years. The cost of drug withdrawn from market is huge. Therefore, it is critical to assess DILI liability early in drug discovery in order to de-risk drug candidates and improve the success rate of drug discovery. Clinically, drug-induced liver injury can be classified into intrinsic liver injury and idiosyncratic liver injury. Intrinsic drug reactions are caused by drug-related properties. Drugs that fall into this category cause reproducible injuries in animals, and the injury is dose-dependent. The injury can be due to the drug itself or to a metabolite of the drug. The intrinsic DILI are associated with dose and lipophilicity of the drug. formation of reactive metabolites, formation of reactive oxygen species, the ROS, mitochondrial dysfunction, inhibition of hepatic transporters, and the nuclear receptor FXR that regulate the expression of those transporters, induction of steatosis and phospholipidosis, and inflammatory reactions. The second category of DILI is idiosyncratic DILI. Most idiosyncratic DILI cases happen at the therapeutic dose in an individual, even though this dose is well tolerated in the general population. Idiosyncratic drug reactions are related to the characteristics of individual patients, including demographic factors such as age, gender, and race, genotype, underlying diseases, and co-medications. Idiosyncratic DILI is considered dose-independent. The assays that are currently available are relatively suited for assessing intrinsic DILI risk. It is very challenging, if not impossible, to predict idiosyncratic DILI by in vitro assays at the current state. Because DILI can be a multi-step and multicellular disease process, the prediction of all forms of DILI is inherently impossible by a single assay. In a publication this year in Nature Review Drug Discovery, a group from European pharmaceutical companies and academia partners proposed a roadmap for the development and deployment of in vitro predictive models for DILI. They take an approach contains a tiered system utilizing currently available in vitro assays and integrating with emerging cell-based technologies for DILI assessment. For example, Tier 1 in this roadmap compresses single-cell systems such as using human primary hepatocytes, cell lines, and reporter signaling. Those single-cell systems evaluate immediate chemical biological effects, such as bioactivation, drug or bioacid accumulation due to transporter inhibition, mitochondrial toxicity, and signaling associated with oxidative stress, endoplasmic reticulum stress, and inflammatory signaling. And Tier 2 assessment in this roadmap, a more complex system containing liver cells in a more physiological state, including assays in multi-cell culture, spheroid and 3D culture, long-term culture, and microphysiological system. The aim of the Tier 2 system is to assess long-term effect of drug exposure. And Tier 3 compresses a complex test system with the goal to be able to provide prediction on idiosyncratic DILI. At Eurofins Discoveries, we also take this tiered approach with the aim of managing DILI risk and assisting in the design and the development of safer and more effective medicines. We employ a panel of assays to assess one or multiple factors that have indications to cause DILI. We offer two panels of in vitro assays in order to assess DILI risk at the early stage of the drug discovery process, the DILI screening panel and the DILI mechanistic panel. The assays in DILI screening panel are high throughput and low cost assays. They are suitable for large amounts of compound screening, such as compound library screening, to weed out the obvious offenders. There are three assays in this panel that assess the general cytotoxicity by running a 72-hour cell viability assay in HepG2 cells and assess the mitochondrial toxicity by running cell viability assay in HepG2 cells cultured in medium containing either glucose or galactose. The assays in DILI mechanistic panel can reveal possible reasons that a compound causes DILI. We offer a combination of 6 assays in this panel that assess the general cytotoxicity in human primary hepatocytes, high-content imaging cytotoxicity assay that measures 5 cellular parameters, including cell number, cell membrane integrity, intracellular calcium level, nucleosides, as well as mitochondrial function. And cholestasis is one of the causes of DILI. Inhibition of BSAP, which is the major efflux pump for bile acids from hepatocytes, and the agonist and antagonist of nuclear receptor FXR, which regulate the expression of VSAP, are used to predict cholestasis. The induction of phospholipidosis and glutathione conjugation assay are also included in the panel in order to cover a broad range of the causes of DILI. We use two types of cells in the assays that assess DILI risk: human primary hepatocytes and HepG2 cells. There are advantages and disadvantages of each cell type. Human primary hepatocytes can be either freshly isolated or cryopreserved. The human primary hepatocytes are considered as the gold standard of in vitro hepatotoxicity assays because they are the closest in vitro system to the in vivo condition. They return functional activity of major drug metabolizing enzymes and drug transporters, and a short-term 2D culture or relatively longer term in 3D culture or co-culture with other hepatic cells. But there are some limitations of using human primary hepatocytes, mainly due to phenotypic instability, short lifespan, batch-to-batch variation, which limits the reproducibility of the assay. The cells are significantly more expensive than the HepG2 cells, which also limits its usage in drug screening. And the HepG2 cells are widely used in drug discovery for hepatotoxicity assessment. They are derived from human hepatoma cells. They have the advantages of high stability, unlimited lifespan, ready availability, and high reproducibility. The major limitations of HepG2 cells are that they generally do not express phase 1 and phase 2 drug metabolizing enzyme and drug transporters. Now I'm going to talk about the in vitro assays that assess mitochondrial toxicity. Drug-induced mitochondrial dysfunction is considered as one of the important determining factors in the onset of drug-induced liver injury. Mitochondrial toxicity can be assessed by a set of cell viability assay in which HepG2 cells are cultured in the medium containing either glucose or galactose. When the tumor-derived cells, such as HepG2 cells, cultured in the medium rich in glucose, they generate energy by glycolysis, which bypass the mitochondria. They are resistant to many mitochondrial toxins. However, when the glucose is replaced with galactose, the cells are forced to use mitochondria to produce energy. Therefore, the cells cultured in galactose medium are more susceptible to mitochondrial toxins. It is a commonly used in vitro assay to assess mitochondrial toxicity in drug discovery because it is simple, fast, high throughput, and most importantly, it has high predictivity value. There are many peer-reviewed publications that validate the utility of this assay in identification of the drug caused mitochondrial toxicity. We have validated a high throughput screening assay that by using HepG2 cells cultured in glucose versus galactose medium. In the validation study, we tested 20 reference compounds with 4 compounds that are non-mitochondrial toxins, highlighted in orange in the table. The other compounds do not have mitochondrial toxicity but cause cell death, which are highlighted in gray, and 5 compounds that are not cytotoxicity in this assay, which are highlighted in green. The assay can correctly distinguish the non-mitochondrial toxins from non-mitochondrial toxins by using the IC50 ratio between glucose and galactose, which is presented in the last column of the table. We can see at the right-hand side of the slide the dramatic difference in those dose response curves for mitochondrial toxins. The green plots are the inhibition of cell viability from the cells cultured in galactose medium, and the blue plots are from the glucose medium. The results demonstrate that the assay can be utilized to identify compounds that disrupt mitochondrial function by different mechanisms. For example, FCCP which uncouples mitochondrial oxidative phosphorylation from ATP synthesis, showed almost 20-fold shift of the IC50 in this assay. Antimycin C, which inhibits complex 3 cytochrome C reductase in mitochondria, showed over 1,000-fold change of IC50. Rotenone is a classic mitochondrial inhibitor that inhibits complex 1 NADH ubiquinol oxidoreductase in mitochondria. We observed over 100-fold of IC50 shift between the glucose and the galactose assays. IC50 ratio gives us a clue that if a compound may cause mitochondrial toxicity. Those assays can be run in either 96-well plates or 384-well plates with 5 to 10 business days turnaround time. Those assays can be utilized in early stage of drug discovery because they are predictive, require minimum amount of compounds, have fast turnaround time, and cost effective. We use high-content analysis imaging assays to illustrate the mechanisms of hepatotoxicity caused by test compounds. High-content analysis technology can analyze multiple parameters in the same cell simultaneously by staining the biomarkers with different fluorescent dyes. This cartoon illustrates what happens when healthy cells are undergoing cytotoxic attack. In general, we will see the decrease of cell number due to cell death or inhibition of cell proliferation. We also may observe decrease in nucleosides, which is the result of chromatin condensation, is one of the hallmarks of apoptosis. Decrease in mitochondrial membrane potential, which indicates mitochondrial dysfunction. We also know that the increase of intracellular calcium, membrane permeability, and reactive oxygen species may cause cytotoxicity. By measuring those cellular parameters, we can provide you with mechanistic information on the reasons that test compounds cause cytotoxicity and ultimately DILI. We offer a high-content analysis imaging cytotoxicity panel in HepG2 cells, which measures 5 endpoints simultaneously in individual cells. In this assay, cell viability, nucleosides, mitochondrial function, intracellular free calcium, and cell membrane integrity are visualized by the staining of different fluorescent dyes. As we shown here, the top panel of images was taken from the HepG2 cells that were treated with vehicle control. In this case, is 1% DMSO. The bottom panel of the image was taken from the HepG2 cells that were treated with 120 nanomolar of cerevastatin. We can see the clear differences of each endpoint after 120 nanomolar cerevastatin treatment. And this assay can be run at multiple concentrations to determine IC50 or EC50 of a test compound. After analyzing the images, those response curves can be plotted, and the IC50 and EC50 values can be calculated. As I'm showing in this slide, the effects of cerevastatin on nucleosides, mitochondrial membrane potential, intracellular free calcium, membrane permeability, and cell number are analyzed, and the IC50, EC50 values are reported. You can easily rank your compounds based on the IC50 or EC50 value of each measured parameters. The utilization of high-content analysis imaging assays to predict DILI in humans has been extensively validated, and there are a few publications of those validation studies. For example, in a study in 2006, O'Brien tested 243 compounds in HepG2 cells by high-content analysis imaging cytotoxicity assay. The results show 80% specificity and 93% sensitivity. Xu published his research in 2008, showing 95% specificity and 50% sensitivity by using human primary hepatocytes from over 300 compounds. In 2014, Garcett's research showed 75% specificity and 58% sensitivity using both human primary hepatocytes and HepG2 cells from 144 drugs. The results demonstrated that high-content analysis imaging cytotoxicity studies are useful in the in vitro detection of drugs that have high propensity to cause DILI in vivo. Those assays have the potential to help selection of drugs with reduced likelihood to cause DILI in humans when used to support compound profiling in drug discovery. The high-content analysis imaging assays that we offer are summarized in this slide. Those assays can be run in either HepG2 cells or human primary hepatocytes at any time point from 4 hours to 72 hours. It can measure multiple cellular parameters such as cell number, ROS, mitochondrial membrane potential, intracellular calcium, nucleosides, membrane permeability, and heat shock protein 27, representing cellular stress. Those assays can be customized to meet your specific needs. Now I'm going to talk about the in vitro assays that we use to assess cholestasis. Cholestasis is one of the major causes of DILI. Clinically, cholestasis is defined as the reduction or stoppage of bile flow, which results in the accumulation of bile acids in hepatocytes. Traditionally, the BSAP inhibition assay is used to predict a cholestasis in vivo, but recent research found that the correlation between BSAP inhibition and DILI outcome is not significant. There is a story from two drugs that block BSAP, cholestatic and pioglitazole. Both are type 2 diabetes drugs. Cholestatic was removed from market in 2000 due to DILI caused by a cholestasis. Pioglitazole is still on market and considered as a safe drug to treat diabetes. Pioglitazole is a more potent BSAP blocker than traglutathione, but it does not cause a cholestasis in clinic. So the homeostasis of bile acids is regulated by transporters which transport bile acids in and out of hepatocytes, and CYP7A1, an enzyme that catalyzes bile acid synthesis. So on top of those, a nuclear receptor FXR regulates the expression of those transporters and enzymes. Research found that bile acids serve as a hormone-like signaling molecules to active nuclear receptor FXR, regulating the homeostasis of themselves. When the bile acid level increase in hepatocytes, which may be caused by the inhibition of BSAP, the FXR is activated by the high concentration of bile acid. The activated FXR upregulates the expression of bile acid efflux transporter such as BSAP, MRP2, OST, AMB, and downregulates the uptake transporter NTCP of bile acid, and also downregulates the enzyme CYP7A1, which catalyzes the synthesis of bile acid in hepatocytes. And this way, the homeostasis of the bile acid is maintained even though the clearance of bile acid by BSAP is blocked. Inhibition of BSAP alone may not cause cholestasis. It is discovered that traglutathione is also an antagonist of FXR. Therefore, it inhibits not only BSAP, but also the feedback compensation regulatory pathway. Pioglitazole is not an antagonist of FXR, and this explains why traglutathione causes a cholestasis, but pioglitazole does not cause a cholestasis. We have validated the BSAP inhibition and the FXR antagonist and agonist assay at Eurofins Discovery. We use inside-out vesicles from HEC cells that overexpress in human BSAP. In this assay, tritium-labeled taurocholate is used as substrate. By combining the BSAP inhibition and FXR agonist and antagonist assay, we can provide a better assessment on cholestasis. And this is a summary of the in vitro assay that we use to assess DILI risk. We offer two panels of assay, DILI screening panel and DILI magnetic panel. The assays in DILI screening panel are high throughput, low cost, fast turnaround assays that can be used at early stage of drug discovery to screen large amounts of compounds. In this panel, we use HepG2 cells to assess cell viability after 72-hour incubation, and we use two cell viability assay to assess mitochondrial toxicity. We offer the combination of 6 assays in DILI magnetic panel in order to provide magnetic information on the causes of DILI. Those assays include high content analysis imaging cytotoxicity assays in HepG2 or human primary hepatocytes, BSAP inhibition, and FXR agonist and antagonist assay that assess cholestasis, also phospholipidosis and glutathione conjugation assays. By offering the two panel of the assays, we can provide our clients with a useful tool to de-risk their drug candidates' DILI liability at the early stage of drug discovery. This concludes my presentation. Thank you for your time.
Thank you, Yong, for your excellent presentation today. I think you provided a great overview of DILI and the impact that DILI has on drug attrition. I think you also provided a really wonderful overview of some of the available in vitro toxicology assays, as well as tools that can be used to assess potential hepatotoxicity, as well as the tools that are available at Eurofins Discovery to help clients reduce drug attrition due to drug-induced liver injury. Again, I would like to thank all of you for joining the webinar today, and please feel free to contact us if you have any questions about any of these assays. Thank you again, and have a wonderful day.
本日はユーロフィンディスカバリースポンサーセッションにご参加いただき、誠にありがとうございました。ご不明な点やご質問などがございましたら、画面に表示されております E メールアドレスまでお気軽にお問い合わせください。よろしくお願いいたします。